Thank you, Bert. Thank you, everybody, for giving me the opportunity to talk about Homology Medicines this morning. So I'm just going to walk you through a couple slides about Homology Medicines. We are a publicly traded company. These are forward-looking statements for everyone to evaluate. One of the things about Homology Medicines is we're characterizing and developing a platform of 15 new AAV serotypes. And these were discovered in normal human volunteers for hematopoietic stem cells. That's why we call them AAV HSCs. And what we've done as a company is we've actually worked on really differentiating this family of 15 AAV HSCs. They have very unique properties. What we found is that as small as one amino acid difference in these AAV HSCs can result in very different tropism in non-human primates as we've looked through that. So they're highly efficient, and they also have this unique ability to not only be used for traditional gene therapy using a promoter, but we do see elevations in non-nuclease mediated homologous recombination. I'll talk a little bit about that. We have a very experienced team in rare disease. This has been our main focus. Collectively, I think we probably have over close to 100 years in rare disease, human genetic type research collectively with the team. One of the things that we really take a lot of pride in is moving from discovery to clinic as quickly as we can. So one of the things we've looked at when we started the company back in early 2016 were to take these new AAV HSCs and develop them and show that we can manufacture them, we can deliver them to the appropriate tissue, and we can execute on moving a program to the clinic. And so we expect to be in the clinic in 2019 with clinical data in adult PKU with our lead program. And then finally, we have a broad intellectual property estate covering the 15 AAVs, composition of matter, but then in addition to that, the high efficiency and the non-nuclease-based editing within the clade that these AAVs map to. This is just a slide that highlights a little bit of the subtleties within the platform that we're developing. So we call ourselves a genetic medicines company because we can look at the biology of a disease and then assess how we want to tackle that with our AAVs. If it's a disease where the cells are very slowly turning over or not turning over at all, such as either the adult liver or the CNS, we can utilize the AAVs, as you see on the left side of this slide, to do a traditional gene therapy where we'll engineer a promoter to express the transgene of interest and deliver it to the cell. It takes it to the nucleus, and then we get expression off this episome that doesn't integrate into the genome. On the right, for diseases that we want to tackle that are rapidly dividing cells, such as a pediatric population, so a very young child, if we want to go into the liver, that liver's going to expand. We want to make sure that we integrate into the genome such as those cells divide that allele thinks it's making a mutated form of pH, but it's actually making a fully functional phenylalanine hydroxylase. Taking that, this is our portfolio. Our lead program in PKU will be in the clinic this year. We expect data readout by the end of this year. We also have two programs in IND enabling. Our first CNS program is in a disease called metachromatic leukodystrophy. It's a very aggressive lysosomal storage disorder. It takes advantage of the fact that these AAVs can cross the blood-brain barrier and then hit different cells that are known to be secretory in the brain. And then finally, our pediatric PKU program. We're trying to target the adult population where we can integrate the corrective gene into the liver, and then as that liver divides, we can retain that expression over time. This is just a little bit of preclinical data that we've generated on the PKU, our lead program. And what you'll see in this, there's a mouse model. It's called ENU2. This model has a mutation within the mouse version of the gene for PKU. It's called phenylalanine hydroxylase. When this gene is mutated on both copies, These mice can take in phenylalanine, but they cannot metabolize it. It's an essential amino acid. If you look on the left side of that bar, you'll notice at the very beginning, you'll see that the phenylalanine levels of the serum are as high as 1,500. After a single dose in this model, we restore that to normal levels, and then we've seen that throughout the life of this particular model. This model's on the background of BTBR, and it lives approximately about a year, and so we've restored that over that course of time. In addition, expressed in the liver. It's a cytoplasmic protein. It sits inside the cell of the liver and metabolizes go down. Is reflected in the showing this restoration of this phenylalanine to tyrosine ratio, which is also part of the clinical diagnostic and management for the patients with PKU. And then finally, I'm going to leave you on this. We've just recently finished our new GMP manufacturing process. It's 25,000 square feet. We're at 500, where it's built for 500 liter scale. And this is a dual process that we can manufacture either our gene therapy or our gene editing programs all the way through fill finish. And so this now will be really fully functional as we move into this year and supporting our programs as we move them into phase 1-2. We felt as a company it was very important for us to control the whole process from process development all the way through fill finish under our roof and in our control.
Thanks. Thank you, Albert. Come on over. Love to talk a little bit more about your technology in the context of some of the discussion with regard to off-target activity of nuclease-based approaches. Could you talk to the precision that you're able to affect both on-target activity as well as off-target activity? How do you measure that? What data do you have that shows you're able to affect things appropriately?
Yeah, it's a great question, Bert. So one of the processes that AAVs utilize and how they integrate into the genome is a process called homologous recombination. This is the highest fidelity, highest precise natural cellular mechanism that's used for DNA repair. What we found is that AAV, and this was originally discovered in the 1990s, that AAV, when you have a single-stranded genome, can evoke this particular process, homologous recombination. It's what a lot of people out there, even in the nuclease world, is trying to do because of that precision of integration. By taking advantage of that, it gives us this unique ability to put a gene into the genome. And so by utilizing homologous recombination, we've been focusing our efforts on taking a gene that's missing and specifically inserting it into the genome. And because of the fidelity of homologous recombination, if we put something into the genome that we want to have expression off of that, we need to make sure that there are no unwanted mutations that could cause a frame shift and actually reduce the expression of something that we're putting in. And so what we've been able to do with this homologous recombination-based approach is we'll assess it in two ways. An on-target approach, where we'll integrate into a region into the genome that we plan to, and then what we do is we do next-generation sequencing, very deep sequencing, across the DNA that was extracted from those treated cells or treated tissue in the mouse model. And then we sequence across that at more than 10,000-fold deep at each particular nucleotide to assess at each spot, what do we see only the intended change that we wanted to make, or do we see other changes? And that's where that's the process of homologous recombination is very high fidelity. We're not seeing any unwanted variants that we want to put in. For the off-target, we do a similar thing, and that because we're putting something into the genome, we can use that. So for example, the PKU program, we're putting phenylalanine hydroxylase into the genome. We can take that cDNA or that functional copy that's in the genome, and now instead of just sequencing across that, we can use that once we extract DNA, and then we sequence out, and we use bioinformatic approaches to say, where did it go? So in a sense, it's unbiased in the sense that we're not asking or making predictions of where it may go. We're actually just taking the treated samples and seeing, and again there, we see very precise on-target as well as off-target profiles with the programs.
there have been other technologies that have been able to affect homologous recombination, yet yours seems to do it with a much higher efficiency. What's happening biologically to affect that efficiency?
Yeah, so what we know is that with AAVs in general, and I think Bert's referring to some of the earlier work where you saw this tremendous increase in efficiency from what people were doing. They were using, say, double-stranded DNA and getting efficiencies of maybe 1 in 10,000 cells or 1 in 100,000 cells. Once you moved to AAV, it was found that you could get up around 1%. What we found with this clade that we're utilizing is now we can go in from 1%, we go up another log. So we're at the 10 to 15% levels of efficiency of integration. And what we know and what we've been able to characterize is that once these AAVs bind to the cell, they take that single-stranded component that's in there to the nucleus very efficiently. So we see several orders of magnitude quicker and more efficient approaches of getting that single-stranded DNA into the nucleus that then drives the homologous recombination in that process.
I'd like to shift to manufacturing. You talked about it at the end of the presentation. Do the vectors that you're utilizing provide advantages? And you've chosen to bring a considerable amount of that capability in-house. Talk about that a little bit more.
Yeah, absolutely. So one of the aspects about having 15 of these different serotypes, that's one thing that we also evaluate across each one of them. We call it the manufacturability of that. So we'll look at how do they biodistribute throughout the body, whether it's in a non-human primate. We can then assess that, and then we start to look at how well do they actually package and produce. And so across these 15, you see variability in that. Some package extremely well, others still package well, may not. So we build that manufacturability in very early. The other thing that we built this right from the beginning is a very strong process development team. And so they'll start to work on upstream, downstream processes that we then will tech transfer to our external partners for this. But then as we were going through that, bringing in the expertise of my colleague, his name is Tim Kelly, who led technical operations at Shire, bringing in all that experience of having it in-house, then really gives us the flexibility as a company to take that same process development work that we were doing and then giving it to external manufacturers to then do that same kind of tech transfer internally, and allows us to then be in control of our queue. So as we're working to manufacture and working towards bringing our programs into phase 1-2, we're working on producing material for our GLP tox studies. We can then make those shifts as a company, and it gives us a lot more flexibility in deciding that queue. But then it also allows us internally under our roof to establish and assess that quality all the way through the process. And so that's one of the reasons we felt it very important to do that, because I think that's one area that a lot of companies can stumble on, and that is manufacturing. You know, where the AVs are great at working in a mouse, but unless you can make them at scale and at quality to move into the clinic consistently every single time, that's what you really need as a company. We felt that was such an important aspect of what we want to deliver that we decided to make that investment internally on that.
Thanks. I'd love to shift to the clinical program. PKU, why was that your first choice with this technology broadly? And then could you talk about what you've seen in terms of tyrosine levels and what that might mean clinically for these patients?
Yeah, absolutely. So when we were setting out the company, we were looking at these novel AAVs. So they were discovered naturally in normal human CD34 cells. However, they were never taken forward as a therapeutic, as a recombinant AAV. So they are novel on that front. So when we were looking and setting up the company portfolio, we thought our quickest route to understand the properties of these AAVs was to do a traditional gene therapy approach. With that in mind, we then looked through the landscape of different disease opportunities, and we landed on PKU for a number of reasons. There's a significant unmet medical need within the adult PKU population that's currently unmanaged, whether it's with diet or even some of the newer therapies out there, because it's not reestablishing the normal biochemical pathway. Number two, it's a liver-mediated disease, and so we knew we could target the liver with these AAVs. And number three, because it's an adult population, we felt that we could get long-term durability from a treatment, a single treatment in the adult PKU population that could potentially allow them to come off diet. So for all of those reasons, we strategically chose PKU for that lead. We've learned a tremendous amount about our AAV HSC15. We learned manufacturing. We learned a lot of safety. We just learned a lot of characteristics and properties of that particular molecule. That's the same capsid then that we can then bring to our editing. So working in PKU, a very well-established regulatory and clinical path. Now we have safety on that marker. We can then apply that to our pediatric population, and now we'll add the new part, which would be the gene editing. So we've already established all the other aspects of moving it forward, and then we just add on the newer component, which would be the in vivo gene editing. So that's why we picked PKU.
Okay. And we're getting right to the end. Just a quick question. You're on track for the clinic, and there will be data in 2019?
There will be data in 2019, yes.
Terrific. Thank you very much, Albert.
Yeah.